10 o'clock and I started the recording.
Great. Hello, everyone. We're going to wait a minute or two while we wait for additional people to join the call. So we'll be starting in just a sec. Okay, I'm going to start this webinar. I want to thank everyone for joining us. I know a lot of people will be joining us, joining at points in time in this webinar, and we're also going to be recording the webinar so other people can see the webinar afterwards. I'll introduce myself. My name is Bill Thompson. I work in the CDC, the United States CDC Division of Viral Hepatitis. I just want everyone to know everyone will be on mute except for Dr. Rowe, our IT person, Kevin Borden, and myself. I will attempt to field questions, and I will just say this is a new normal. So I'm sitting in our basement in my son's recording studio where it's the only computer with a Ethernet connection. And before I introduce Dr. Rowe, let me make a few introductory comments. I've been trying to organize this presentation for the last 12 months. So we were planning this before, I'll just say before things changed. For those who don't know, at the CDC, many of us are deployed to work on the COVID response. So about a third of my division is deployed. About a third of us are working on studies, and another third are pulling their hair out trying to get all the work done. So that's the situation we're currently in. So let me just, in terms of introductory comments, because it's going to be a modeling talk and there's going to be many non-modelers listening, I'm just going to say an important thing happened recently where the CDC made viral hepatitis a winnable battle. And that's a really big deal because it leads to more investment of resources to eliminate viral hepatitis, and in particular B and C. So it's becoming more important, and I will say once things settle down from the COVID response, the CDC will invest more time and energy in some of these winnable battles that's difficult to do right now. Second thing is yesterday was hepatitis testing day in the United States, May 19th, so it's appropriate that Arni should be giving a talk this week. It's hepatitis month as well. So if you haven't been tested for hepatitis C in particular, I would suggest you get tested. The last thing is just to introduce, before I introduce Dr. Rowe, is that we do value modeling at the CDC, and it's a challenge. There's this tug, between the, let's just say, CDC epidemiologists and modelers, because the modelers bring new innovative ideas to the table, and we are constantly trying to figure out how to incorporate that information into our daily work. So this is an example of Lin Zhu, who is currently at Stanford working under Josh Solomon, doing some HCV modeling for us. And this, for me, is just an example of really valuable work where on the left-hand side, you see a picture that describes a network of injection drug use modelers, and not modelers, individuals, and the modelers are modeling the data. And the bigger one is really what we think is the real description of the network. So the value of modeling, from my perspective, is something like this, where we say, we can't rely on this single sample, and we want to think of it as a bigger network of individuals and to think about the assumptions we're making. So I just think it's a really good example. And then Dr. Rowe wrote a recent commentary that he'll mention this work as well. So let me introduce Arni. So Arni is a tenured full professor at the Medical College of Georgia and the director for the Laboratory for Theory and Mathematical Modeling at Augusta University, Georgia. Prior to this, he taught and performed research at several premier institutions, including the Indian Statistical Institute, the Indian Institute of Science, University of Oxford, England, and University of Guelph, Canada. Dr. Rowe's mathematical modeling and deep learning AI research has attracted more than 30 media coverages during the past 7 to 8 years, and has worked. He has appeared in Research Excellent Framework of UK in 2014. He received the Heiwa Nakajima Award from Japan and DST Fast Track Young Scientist Fellowship in Mathematical Science from New Delhi, where he performed work at ISI, which is the Indian Institute of Science. So Dr. Rowe has been principal investigator and co-investigator or joint investigator on numerous international and nationally funded grants and studies. And I'll just say is well published, and we are thrilled to have him here today. And I will just say the title of the talk is Innovations in Mathematical Modeling for Describing Epidemics and Providing Time-Sensitive Recommendations, Experiences from HCV, HIV, and COVID-19. With that, Dr. Rowe, I will turn it over to you. All right, Arni, let's see if we can pull this off, transitioning. There we go.
Yeah, thank you, Dr. Bill Thompson. I'm really honored to get introduced by you. And I want to thank the CDC for inviting me to give this webinar. So like many other academicians, I also for the last 15, 20 years, have been teaching like a pure mathematical courses like differential equations, real analysis, complex analysis, and also applied courses in the, like a stochastic process, population biology, many courses. But then I do have always desire to see that the mathematics, how the mathematics, what we do in academics, how that is practically being applied. I was very curious for that for the many years, and I was really lucky to get interacted with through that process, very leading the academicians across the world, senior administrators in the governments, politicians who are actually those politicians who are actually publicly elected politicians during the process of policy making. I'm really lucky to be working with several people over the last 2 decades. So that's why when the CDC asked me, I felt so happy to explain all these things, and they also asked me not to just speak on COVID-19, for which we are really busy. They just want to go through the other modeling also, what we have been doing. So in this connection, first I would like to dedicate this talk to Dr. Robert May, who has been my mentor, and he worked with governments, and then he helped with policymakers, and then unfortunately he died last month at the age of 85. So I would like to dedicate this talk in the memory of Bob May. So Bob was my good friend, and I wish him rest in peace. And then the organization of the talk. The organization of the talk, if you see that, then that organization, first I will start with new approaches in HCV modeling. And then I will discuss the HIV modeling success stories in India, and then the new models for poultry, bird diseases, and then I would end up with COVID-19 models. I'd end up with COVID-19 models. So first, let us start with the HCV modeling. So in general, the questions asked in HCV are, which individuals to target for prevention of HCV, and how do we model the behavior of the injection drug users. So these are the general questions have been asked by hepatitis C. HCV stands for hepatitis C virus. How does these two, how to model the behavior of injection drug users and then the individuals, whom to target. And then so far the modeling has been informative to date in the following aspects. Modeling help to understand the community-based transmissions of hepatitis C virus. And then models also helpful for, regression models in predicting the sero status of individuals in the population, and the deterministic models, and also agent-based models. And then the key literature, in recent years I found very attractive was Tricky et al., which Dr. Thompson just mentioned. So I wrote an article, the comment, based on the, the work done by this article. And then NM Dikshit et al., I see that, they do a lot of simulation work, very, very helpful, useful. And then the Natasha Martin, I see that, she also published a brilliant chapter recently in the Handbook of Statistics. And then the Belrose, they wrote a review article very recently. And Panda et al., they also worked on the HCV among the injection drug use. And Sood et al. Worked with the brilliant data in various population-based surveys in India, in Punjab. So I would like to thank all these authors, write brilliant articles and inspire me to investigate the questions within hepatitis C virus. And then when I found these articles, I thought, just use the, some of the common senses, because I've been working on mathematical modeling and then, various probabilistic approaches and the network grid models. I thought, why not we try to, because the aim is to reach the individuals who need the, the individual, really target the individual, if the seronegative individual, to protect him not to get infection. If it is seropositive, so that, we make sure that, that individual do not transmit the virus to others. So the proposed new hypothesis, what I proposed recently, last year and then published this year, is that, the allocating probabilities, the probable distribution to seropositive individuals and repeated simulations might not be adequate for clustering of population. The clustering is very, very seriously studied by all the hepatitis C modelers. And then the allocating the probable distributions is not sufficient, that way is not sufficient. Because one of the important preliminary tasks for formulating epidemic control program intervention is to identification of clusters that drive an epidemic in a population. Even if you assume the survey data and corresponding weights are accurate, which are as accurate as possible, the data collected might not be sufficient to optimally identify the population. Hence, this new hypothesis we want to test on the population that just appeared in the epidemiology infection. So the new method works as follows. So the new proposed idea is to first, we create a graph with n number of vertices and m number of edges. And then once a graph is constructed, then within each node, various attributes are measured, like the demographic, clinical, social, medical, many other factors, genetic, many other levels of attributes have been collected at each node. A node in a sense vertices, vertices and node are same. So that out of all these n vertices, these additional information on attributes are being collected, and then the edges are collected, and after that data collection done, then the edges are connected according to the attribute level data. So this is the novel idea introduced. The originality, that's what the originality is, that to bring the graph structure to store the attribute level information. That's what and then I have a technical slide, but I can come back to the technical slide at the end of the talk, if there are question answers, but I will be happy. As I mentioned, this will be a non-technical talk. So the new proposed idea in terms of the visualization, suppose we have a graph structure like that. We form from the raw data, as I mentioned, the brilliant studies there. So suppose we take the data from those studies and then formulate a network graph like that. And then here the red color, those circles indicate the seropositive individuals, and the white circles are those individuals who are seronegative for hepatitis C. And then that's how they share the needles. They share the needles, the network. This is an example network. It's not this is just a, for example, like this is not a real network from any data. This is an example network. And then this network, now we break this network into smaller subgraphs based on the attribute level information as I just explained. And then we have also, developed algorithm to how do we break this into smaller subgraphs. This is called, a graph and a subgraph. So then at each node, the red color circle, you see that there is cylinder here. So that node, the cylinder level information is captured at each node. That's what the originality here. And then breaking, once you break it, then you introduce the attribute level information. And that information which is collected at the attribute level, both for the seronegative and seropositive, and then the network is studied based on the attribute information, and then we target them for the intervention. Because we know that regression models are very powerful, no doubt about that. They're very powerful statistical tools, but they could at most give you an average sense of the regression models might tell particular attribute is associated with the injection drug users. That means we target all the individuals who do not have hepatitis C or who do not have the seropositive status, but then because regression models are give a general average sense of the information, they might not give us the particular information for that particular individual. Hence, the regression models, all those, they are very powerful tools. At most give an average sense, and then for optimal clustering, such models could be less effective. That's what, suppose we take the two graphs, G1 and G2, I'll go a non-technical way, constructed in two population surveys. Suppose G1 is from one population survey, you construct a graph, and G2, another graph constructed by another population survey. And then these two graphs, how close they are, they are similar, how similar they are. That is what, the isomorphism, that's what we try to understand, from the one population we construct and then we match. So from the matching algorithms also widely available, one of the matching algorithm I personally liked was, Bonici, they publish in BMC Bioinformatics, this article, so such algorithms, can be explained, these can efficiently match. Suppose we have the graphs I showed you, the synthetic graphs. So those kind of graphs can be compared, and then can be seen that how two different populations, two different graphs emerge from the two different populations can be compared. So this comparison can be done through the matching algorithms, and then once the matching algorithm is done, and then we target the intervention. So that means, what did we achieve? That's what, if you understand broadly, what did we achieve from this new method? First of all, we are able to provide deep target interventions, because those individuals who are seropositive, we can directly target to them with a treatment, and then how to protect themselves, like sharing the needles, how not certain things they should not do. So we are really reaching an individual here rather than a population which has similar characteristics. And then this avoids the misuse of resources, because when we target intervention, the resources of, we don't have to provide the facilities for those individuals who do not have risky behavior, although, they're treated as a general, that particular population could be at high risk, but then the individual may not be at high risk. So we can avoid. So this new type of experiments now, we would like to do with CDC to see that how these new techniques are applied at the ground level, And then the impact, other people, when I published this article on the new approach, then I really want to thank these authors. They said that adding attribute level information to each vertex as described in the letter by Rao is interesting development to these network structures. And these people have been working into the, like Dr. Thompson, other people who have been working tricky, and, they have a group of like Martin, they all been working into the hepatitis C for over a decade. So I felt this is very encouraging, to see that, the new methods have been appreciated. And then they also say that we advise those designing future studies among people with injection drug use in this setting to try account for social network structure and apply the methods outlined in the letter by Rao. So that means these new things they are going to implement. That's a big boost for the mathematical methods. How best we can take the new methods to the ground level to eventually target the hepatitis C. We want to eliminate hepatitis C. We want to see that this is possible. So additional approaches proposed, other than the what these have been published, some new techniques also we have been working. So one of that is to build deep learning networks over the large network data, deep learning algorithms over large network data. I mean, if you have a large network data, we want to develop some new methods of, identifying the individual, training the machine for that, and then using these algorithms, what we developed, and then taking those new algorithms to the targeted intervention, more deep-rooted targeted interventions. So I will briefly also describe the new method here. Suppose we have on the left side, you have the network observed at time t1, and on the right side you have a network observed after some time, after a few weeks, and you see that the black dots, black circles, and some of them are, and the white circles, seropositive, seronegative. So some of the seropositive individuals could have recovered, some of them not recovered, some of them actually negative became positive, some of them converted into transmission already occurred. So this is real data at 2 time points. Now suppose say based on the network observer T1, some of the modelers also try to project the future network transmission, the future, how the network looks like in the future. That means the dynamics of the network they study. So that means they consider a probability distribution, and then they take a random variable, and then they study the transition, how this transition of an individual who is negative, seronegative would become seropositive, or seropositive becomes seronegative, or remain the same. So then we see that the projected network based on some random distribution look like that. Whereas the real data, as we just saw at time t2, looks like that. That means even though we develop some, this is based on stochastic networks. I mean, some of the articles are working stochastic, stochastic models, and then they are trying to build the, the probability, whenever the probabilistic, these sort of changes of states are there. That means this is based on the stochastic algorithms. And we see that still even though people are building stochastic models, there is a, that might not able to predict accurately who would be the positive, who would be negative individuals in the future. Hence, the deep learning algorithms, what we're currently working, might able to overcome this particular limitation also. So the networks are better than regression and the regression models in general, and then these networks can be improved further using the deep learning algorithms. So then, once this HCV work is done, I would like to explain to the success story in HIV modeling done for Indian data, and then how do we create how does that create an impact into the policy. So the key collaborators in this project was Dr. K. Sudhakar from New Delhi, and who has been a team leader and into the he's into the government policy committee in the government of India there in India. And then he really, he's a public health expert. And then he and another his colleague, he's also national committee member for the understanding AIDS policy, Dr. K. Thomas from Pondicherry. So both were actually national policy makers for HIV, their policy committee. So they asked me to whether I can because earlier myself and Dr. Sudhakar, I was working in his team for the World Bank when I was working at the World Bank. So they know the impact of mathematical modeling. So it is their greatness, they try to hire a mathematician to develop mathematical models for, practical. I mean, I have been publishing the academic type of work, but then the bringing the academic type of work to the policy level, that's what they were interested to, see that how the mathematical models are being applied and developed. And then the team was joined by Dr. R. Bhatt from Mumbai. He's a management professional. He was at Indian Institute of Management, and then uh, like they have been trying to build. Trying to use the mathematical models what we are building. We build back and forth together, and then because when they approached me, I was at the University of Oxford, so my host, Dr. Maini, Philip Maini, he was faculty there. He was never been working on any epidemiology problems, but I know because I was there, I requested, whether he can join me, so that although the project was asked for, project people have asked me to develop. Then the major article, the first major, among all the articles, this is the impact, that came in the American Math Society's notices in 2012 on this is the second line, this first model for understanding the second line treatment for India. And then the other major articles, the earliest article came in the Current Science, and then the Lancet, and then also, many articles, but then there are some of the key articles, Then we also published in Springer. They invited us to write a summary of how did we achieve the HIV numbers reduction, what was the role of mathematical models. So that particular article influencing HIV/AIDS policy in India through mathematical modeling, that also we have published the article. And then these are steps for some of the few articles, which are, Current Science is one of the very, very leading journal, in the science. So I appreciate, I thank all those journals, who are very cooperative to, I want to thank all of them. And then the aims of HIV study, I'm also not going very technical details of this. All the articles are available in journals. So the main idea was to provide the third round estimates for the HIV/AIDS, estimates for the third round national AIDS policy, 2006 to 2011. And then the fourth round national AIDS policy, 2012 to 2017. And then we used, we developed just one such example model. I will not go details of this model. I can keep that to the end, to just any questions are there, I can answer those. But this sort of, example models. And then what did we do newly for that is we brought all because I was partly working with the government planning committee there, it helped me to really see all the targets, what are the targets government is want to implement before the project being launched, third phase of policy, and all the variables and the parameters, some of the parameters we estimate from the raw data, some of the parameters obtained from literature. So all the detailed variable, it is not that I develop an academic model sitting in my office, no. So we had a back and forth sitting meetings between between various policymakers. As I mentioned, working with the various politicians, various level of implementation is done, various levels of what exactly the policy, which part of the policy to be targeted. So we divided the entire policy into three phases, like 50% of the targets reached, 75% of the targets are reached, or 100% of targets reached. So if different levels of targets are reached, how that would affect the HIV status in the country. That's what our aim. And broadly, this is the schematic structure. You see that various populations, this is called transmission dynamics structure. Like various populations interact with various other populations, and then we consider by age, we consider by the gender and the various states of HIV. Some of them are susceptible, some they have some of them have STIs, other transmitted sexual transmitted infections, and then this is what the broad picture of the main model. And then the model predictions are submitted to the government in the first phase, that is the first time the government, for the third phase of policy, the report was submitted in 2006, 2007. And then when 2006, 2007, when we submitted the report for the next 5 years, that's how this was looked like. This is what the submitted report, submitted predictions with the blue line indicating the 50%, if the government implement 50% of the targets, how many HIV will be there? 75% target, 100% target. If they reach their own target, that's how the impact of how their target interventions are impacting. And then when we submitted in 2006-7, and when actual data was collected by the health ministry, and then they found that using their own mechanism for seroprevalence at national level, they found that 2009 the HIV estimate was around 2.4 million, roughly 2.4 million. Which is the magnitude wise very large, but then the national, the fraction of population, if you consider with this thing, that's not that, the national average, it's less than 1% among the risk people, So that means that, this is just above the 50% of target interventions by 2009. And by 2011, if you see that, the same thing, it is close to 50%. That means by 2011, government already reached 50% of the targets. That's a huge success. And then, the leading magazine for the Indian Institute of Science, they wrote that, in their one of their investigations, they said that Rao led this effort by collaborating with the colleagues in India and the UK to build a mathematical model. And then they also mentioned that they reviewed our this thing and said that by achieving 50% target with antiretroviral treatment, along with prevention, care and support, the government could bring down the number of people living with HIV from 2.4 million in 2006 to 2.08 million in 2011. And then they say that this prediction turned out to be close. In 2011, the infection burden was brought down to 2.08 million in individuals at that time. And the current science, in their editorial by some authors, two authors in the guest editorial, they wrote that amongst the other steps, the AIDS Planning Committee in 2006 commissioned mathematical modeler Arni Rao to make recommendations regarding feasible prevention strategies. That means the mathematical models have been trusted by the policymakers. That's what more important. It's not that we go and tell them that this is important. It is they believed it is possible. That's what the credit goes to them. They believe that mathematical models can be possible to practically being used. So I felt so lucky to be involved with like hepatitis C or HIV. You will see some other studies also, where the policymaker, it is their role to believe that this sort of techniques can be used. That is what that's what very important, to convince them practically how can we do it. And then also the University of Oxford, they released a press statement. They said that with a research excellence framework, what that happens once in 10 years in every UK university, because I was there that time, coincidentally in Oxford, I was working, I was in the London Mathematical Society Fellowship there. So that at that time, coincidentally, I was there. So I implemented this project from there. And then I came back to India, and when I started as a faculty job there in Calcutta, at that time, I continued the rest of the project. So as part of so that's why they wrote in their impact study, Oxford University, in their impact study, they published, this particular report in their, the press release. So as part of the planning for a national AIDS control policy third phase, Arni Rao was commissioned by members of the NACP planning team to provide predictions of the effectiveness of various HIV/AIDS strategies. This is what they published 13. Like that, there are 13 research impact studies were reported by Oxford across all the departments. So this one came from, this is the only one from mathematics department, out of 13 studies across the university for the last 10 years, for the 10 years period from 2004 to 2014. And then also further, I would not go technicalities, the antiretroviral therapy, we also using the models, we showed how do we understand within the population, how many people how many HIV infected people in the population who require second line therapy. Because the first line therapy, when they were using, there could be some resistance for those individuals, those HIV individuals, people living with HIV. So the treatment would not work anymore on them because they already have resistance on them. So when to switch? So we also using the mathematical model assisted the government, to make them visualize, what fraction of the first line therapy need to be switched to second line therapy. So that also, this is just a summary graph of that, this thing. So with that, I will end the HIV section of this thing study, and then I will proceed to the what we have done for the mathematical modeling of poultry bird infections. Because back in India, I was working on the avian influenza, other things, and then in the UK there is a college called Royal Veterinary College. They have veterinary faculty. They work in various practically relevant projects. And then when they approached the British Council, in India, they suggested, they recommended Indian modelers, and that's how I was being involved in the project. And we won the big grant for the BBSRC, huge grant. And then part of the grant also I implemented while I was in MCG. So we published also using the, I'll come to describe the work, but this also came up in the leading journals, Mathematical Methods in Applied Sciences, PNAS, International Journal of Parasitology, and then, the Veterinary Parasitology. So all these journals, we also showed the impact of models and then the various techniques on how do we understand the spread. So little bit of background, if you see that, the parasites, some of the parasites, they're very dangerous for chicken. They don't kill the parasite chicken, but, they make for example, Imeria is a parasite. This Imeria, when it is infected when it infects the chicken, the healthy chicken, this healthy chicken becomes a sick chicken, they become very sick, Then the sick chicken, they are not profitable for farmers. So they sit in the cage, it's very difficult for them. They don't die, and they have diarrhea, and these chicken, they really, they take 10-day delayed growth, So the delayed growth globally causing, multiple $billion of loss for poultry farmers alone. So that's why, this particular project, we were in that project, the fund where we basically develop a vaccine, and then part of the side project of the work, because as a mathematician involved into the building mathematical models to understand, the impact of vaccine, the heterogeneity of this particular species, etc. And how the drugs and vaccines can intervene into the not happening with the sick birds. Can we prevent them? That's how this particular project evolved. This is how this look like in the lab. Imeria maxima, Imeria tenella, they are the parasites. And then, so if the chicken get infected, what do we do? Do we really keep chicken sheltered in place? What do we do? Or do we just make them make a party like a coronavirus? Or we sit them sheltered, all the chicken, we keep them sheltered when they are infected, or just, we make them just live like they are, just, socialize them normally. Which one do we prefer? So it's not easy, when we keep the chicken leave the chicken freely, it infects other chicken. It go within the pen or within the cage, it doesn't sit alone, it infects other chicken. They share food, they share water. So then how do we help the farmers, what technology we can develop? So I was seriously, involved in that particular project, and then, developed a mathematical model using grid model, And using the grid model, what did we do was we try to understand the walking pattern of the chicken. And then from the walking pattern, the way the chicken walks, not whether it's wobbly walking, but the movement of chicken. Can the movement of chicken within a cage or within a pen, can that help us to understand the density of infections within the particular cage, or how many chicken are infected, or what fraction of chicken are infected? That's what the question we asked. And then mathematically we try to understand, we build a model, myself and D. Blake and F. Tomley. They were veterinary faculty in the Royal Veterinary College. So we published several articles, as you saw there. So one of the article, I picked this graph from there. So the dotted line in this graph is the way, suppose a hypothetical chicken walks in these, the way it walks. And then, but the arrows indicates there are possibilities of chicken going from one location to other location. But suppose a given longest path without coming back to the original location. So that's how the dotted line look like. So we proved several theorems, mathematical theorems also in that article. The various properties of, the NPP problem, and then the understanding how this, but then these particular things, finally help, the farmers, I also presented with the various agriculture universities, these lectures. And then the idea is that, the implications of these theorems, those we proved are relevant to the poultry farmers. That's what we felt very happy, because my collaborators, they are really veterinary doctors. They do have a very great links with the practical, poultry industry. And the USA, as we know that, Georgia produces the largest number of chicken, which supplies, chicken poultry farms and for the entire nation. So in that sense, we want to help the poultry farmers there with the technology. So what we did was using the math grid models. We try to understand how birds walk within the cage, and from the walking pattern, the sick birds walking patterns are different than the normal birds walking pattern. That's what the conclusion there. That's what we brought in the mathematical conclusion, and then we tested on the population. That's what I personally went to I visited several times this Royal Veterinary College. I stayed there, so I did my own, they took permission for me to wear like some special gear to go into these farms. All these things. As a mathematician, I would love to see in reality what's happening without just sitting, handling my own equations. I would like to sit there. So I want to really thank them to make me visualize what's going on there. So then these study, I'm not going to much theoretical, but then these are well received by the poultry community. So one of the leading poultry website that is called Poultry Site by Alice Mitchell, she wrote an interesting article reviewing our work, say that chickens walking pattern hold disease clues. She wrote that. And then MRCVS, this is another leading poultry website in the UK. That author wrote that based on our publication, say that Dr. Rao hopes farmers will one day be able to easily translate walking patterns to identify high risk pens. That's what and then walking patterns could predict disease spread. That's what their title. Another global meat, because meat, this market, the market, like, we have in now Corona, a lot of markets affected. So you remember, all of you, that when the avian influenza came in 2000, about a decade ago, more than a decade ago, how that affected the business. So these all business people are interested to see, the business, the poultry market, they were interested to see how these mathematical models, So we are also developing a new video technology where these, the farmers can feed the bird movements through videos, and then those from using videos, the video, they can give it, the farmers get alerts. Certain birds in certain cages are not well, certain portions of the cages are not well. So that technology also, we are in, several leading, the software industries, they also asked, some time back to see whether they can bring these models can be practically useful. And then also the American Statistical News, they also wrote an article, explaining our work and how that impacted practically meaningful things. And then I will, at the last, I will come to the COVID-19 modeling. So we all know that, the COVID-19 really changed our lifestyles. So that's really, we are having new normal. And then when we saw the cases in January, the cases in US were not that many, maybe a couple of cases in the February beginning. So first thing what I did was, I thought, instead of understanding the spread, I thought, why not we identify who are at risk? Because I looked at some slides by CDC, WHO, all this, they are worried about the growth, but then they're not much focused on the identifying the individual. So I thought, why not I come up with a first model for using the artificial intelligence, because this is like a pathogen is very rapidly spreading. And then I know that, when the pathogen spread, I remember, previous experience of, SARS, or, the influenza, or chicken, the bird flu, avian influenza, the bird flu. I was very much, involved with I went myself to understand the spread. I traveled to the populations with the various, government administrators. So I know so it's important to identify the cases. So first thing I developed was, artificial intelligence model. And then I asked my clinical colleague, if he's interested to look into the work and then clinically relevant. So I want to thank my collaborator, Jose Vasquez. And, then we published this article instantly. So then, I want to thank the journal also who are very supportive. And this is the first AI model, artificial intelligence model for coronavirus detection across the world. And then this has been widely appreciated. And then I want to really thank the various stakeholders who really felt the usefulness of this particular model. And then how this model works. Before I go to the mathematical models for the spread, I would briefly describe the AI model. So at first level, the model collects through an app, collects the symptoms and signs using mobile phones, an online survey, online phone, through the mobile-based survey. And in second phase, thousands of data points can be processed through AI framework that can evaluate individuals and stratify them into no risk, minimal risk, and moderate risk, high risk groups. This is what everything based on the CDC's information online. We took their information online, publicly available data, publicly available information. So at two phases, the collection of the data and then the collected data using the artificial intelligence model that is developed to see that, the data collected, the individual, so that send the individual that particular individual is at risk and go and test for nearest testing location. And that information also will be provided to the customer, to the user. At the same time, the same alert is being also sent to the hospital to which that individual is being asked to visit. So this is the idea of the model. So that way, they know they will be given some sort of a barcode or some identification, so that individual, when they reaches the hospital or the clinic or the hospital or the center wherever the testing is done, that individual goes with this particular alert and say that, this I received so and so, and then they match that, and then that's how this is supposed to work, this AI model. And then that way this simplifies it, because the clinicians, they can't expect thousands of people go to the clinic, and then we know that this how the people are scared. So this model, the AI model was very appreciated, and then the pictorial, if you see that, if you have a geographical region, the data, and then the houses in the region and the individuals in the house. So the mobile-based app, that collects the data, and all may not respond. We know that non-response rates are also very high, sometimes high, sometimes low, but we know that there are people who never respond. So suppose, say, the blue dots explain those individuals who responded, and the yellow circles are those individuals who never responded to the app. And suppose this is what the screen result of the screen test, the app, the model suggests them that these are the individuals receive an alert. The red color circles are the individuals who receive an alert saying that they do have, they might have infection, go and test, go and get confirmation. These app, they are not confirmatory, so app, they will go and test that, you go and test this thing. Then they go and test it, and some of the yellow circles also, you see that, some of the yellow circles, yellow with red, that means they have not responded, but they have symptoms and signs. But then, that's what happens. So these are all, these are legends as I explained. So that's how the AI model captures the individuals who have responded to the survey. And then we also proved, we need to understand what's the efficacy. So suppose there are n individuals in a region, the probability that n1 of those who identified through AI framework are actually out of n individuals are responded. So the probability that identified individual through the framework, given that there are total number of individuals responded to the survey, is n1 n by n square, that also we put in the article. And then we also see that, a lot of reviews by the various industrial, various magazines, industrial magazines. So Fast Company, in California, they wrote, do you have a coronavirus? The scientists are working on an app for that. That's what a very nice review they wrote. Another magazine, Campus Technology, they wrote that coronavirus app to provide at-home risk assessment. So this particular app and the model what we created, this attracted worldwide media. Within 10 days, almost 150 media media houses, they reported this either through video, the news or paper, various things within a short time. And then also, the leading newspaper in India, Times of India, app uses AI to provide at home. And then Jaguar, the leading science writer, Tony Baker, she wrote that app and AI work together to provide rapid at-home assessment of coronavirus risk. And Innovators magazine, they also wrote nice things, Economic Times, and they also like that, various other people reported. Yeah, now we'll go to the various, we have been working, me and my collaborator, Professor S.G. Kranz, from Washington University, St. Louis. He's a top American mathematician, works in complex analysis and partial differential equations. So we know each other, from many years, and he wrote a lot of textbooks, which I followed as a student, so his books are well known. I mean, he's working for the last 50 years, complex analysis and various other things. So we have been collaborating for the last 3-4 years on various other epidemiology modeling, but this particular coronavirus, we have been deeply involved in various things. So one such example I will explain through this talk. So these are the general articles we publish in JTB, Current Science, Infection Control, and medRxiv. Some of the articles are, preprints are available. So one of the studies is on the underreporting. So this is the first study again to report under what level of underreporting of coronavirus across various countries. And we use the wavelets, which is advanced version of Laplace transformations, to visualize the difference between the magnitude of true cases versus the reported cases. So how many cases would have been really there, but how many cases actually reported? That's what we have done based on some data. So as we stated, you all agree that actual pandemic preparedness depends on the true cases in the population, not that whether they are reported or identified. Actual cases, that's what the spread happens, not just based on those cases which are reported. So what did we do? We considered first, the 8 countries data up to the, the March 12th. Before, all these countries except US, they had a first peak by then, and then these are the numbers. So low, that time point, but they were severe, no one expected that time itself. They were, people, no country expected that they will have so many cases by then even. And then the dates within the range, we considered that these are the ranges of dates. And then other factors considered, population density, urbanization, daily new cases, proportion of population, 0 to 14, 15 to 64, 65 plus. And then there was using the math models and other techniques, as I explained, as I mentioned, the available literature. We found that, if we only Wuhan data, if you consider and then apply to the entire China, because entire rest of China, the data was not available at that time point. There could be many more reported, many more cases which are not reported. We don't know that, according to the model. And with the same model, we see that Italy, 1 in 4 only reported by then, March 12th. And then Iran, 1 in 34. And South Korea, 1 in 4. France, 1 in 5. Spain, 1 in 53. Germany, 1 in 3. And then US, actually, the data was, as I mentioned, only 874. And we see that today now more than 1 million. So the data was incomplete and the peak has not reached, but so that is not reliable. Use information, whatever we predicted for US that time. And then we saw that, as I mentioned, wavelets are improvement. So what did we observe is for Italy, for Germany, and for Spain, whatever we observed for the during March first week, by end of the March, that came out to be true. So 3 weeks before the models, whatever predicted, they all came out to be true by end of March. Let us see how that happened. So Italy, what we predicted was, this is what the dotted lines are, the reported, and then the reported versus the actual cases. You see that discrepancy between reported and actual. And see, Germany, they are very close. That means that's why end of the March, the Italy had a big number of cases that emerged, and Germany not many. Although Germany, we already wrote another article. The prevalence, the number of tested in the population highest in Germany. Not the number, but the fraction of, proportion of people tested highest in Germany and next to USA. But the prevalence level in Germany is much lower. And the Spain also, you see that, the reported was much lower than the actual cases. And then end of the March, you see the number of cases in end of March, Spain got emerged. So these 3 countries, the model worked really well. And then, according to our model, there were 90,000 COVID cases, in the US in the middle of March, but only 4,000 were reported. That's why you see that many of those are not reported. They also spread the disease. That's a problem. So we identify cases, those who are, and then report them. But those who are not reported, they also keep on spreading. So unless governments have some mechanism to, increase their surveillance, somehow, randomly increasing has no value. But they have to have some scientifically increase the surveillance so that, they get the true cases. And then we also did for the US, we also predicted the hospitalizations during April to June 2020. And this is with my other colleagues, clinical colleagues, D. Miller and A. Burman, they are cardiologists. And then David Hess, he's our dean, and also he's our neuro professor. So they're all our brilliant academicians and brilliant clinicians. So we collaborated with them to, to understand the, work together, me and Dr. Kranz. So the lower, we gave 3 sets of estimates for the hospitalizations. How many people will need ICU admissions? How many would need hospitalizations? So that's how these ranges of look like. And right now, the US actual data, when you plot it, this week you gave about a month back. This look like, the medium, medium estimate is closer to the, so far what we observed in the May, because we have not reached June, because these projections are from April to June. And then again, the, various, the appreciation from the various, for example, Medical Express, there, Talia, Ogliora, she wrote that mathematical models predict COVID-19 hospitalizations. And then Homeland Security, Homeland Security Wire, they wrote the coronavirus cases. Mathematical modeling draws more accurate picture based on our underreporting study. Augusta Chronicle, Tom Corwin, one of the leading journalists in our area, and he wrote that, the researcher found that thousands of undiagnosed cases of COVID, Augusta area, Augusta Chronicle. And then Business Line, they wrote that India may be detecting 1 in 4 COVID cases. This is not now for the month of March, end of March. That's what they wrote. Business Line is leading science reporter, Jan. And then the Economic Times also, they wrote that, various other news houses, they wrote the impact of this underreporting study. Yeah, so me and Dr. Kranz, we are bringing a special issue for this Journal of Mathematical Analysis and Applications, published by Elsevier. This is one of the oldest mathematical analysis journal, about a 70-year-old journal. It started by Richard Bellman and myself, Dr. Kranz, myself and Joan Nito. We are bringing a special issue on COVID, so that Elsevier are going to announce this soon. So I want to thank my Medical College of Georgia, Augusta University. They have been brilliant. I have a brilliant academicians, colleagues, and the clinicians, staff. We have brilliant staff. They have been extremely cooperative for me to work on the COVID and many other projects. And the students I teach here, the students are really brilliant. I get, they ask very, they come. They take the courses very seriously. When I introduced the new course here, mathematical modeling in epidemics, they received it quite well. I want to thank my students, my PhD students, and the staff, faculty, and then I want to thank our Prof, Dr. Gretchen Kaufman. She has been extremely supportive of basic science research. She herself is a virologist, so I want to thank her. And then I want to thank our dean, Dr. David C. Hess. He's very, very active in promoting the COVID research. He was a really strong supporter of mathematical modeling and the impact. He saw the impact of that. And then I want to thank the entire college. And I want to thank the prior institutions wherever I was there: Indian Institute of Science, Indian Statistical Institute, University of Guelph, Institute of Population Science, Mumbai, and Mathematical Institute, University of Oxford. And from short-term institutes, I was there for several months to a year, Hiroshima University, Japan, Imperial College, London. And the early mentors in biological, Vidya Nanjundaya and Niranjan Joshi, they really inspired me for doing a lot of theoretical biology. They trained me a lot of things. And then the Roy Anderson Imperial College and Bob May, and as I mentioned in the beginning, Anderson May, they wrote a very revolutionary book on disease modeling, and I worked directly with them. That really helped, to visual. They both were close to the government policies, so it helped me to practically daily life, day to day see that, how these models are being taken practically to the there and then back and forth, how they argue. So all these are brilliant. I want to thank them. And then Chris Baugh, I was a postdoc in University of Waterloo that time at Guelph, and Masa Kakehashi also same, in early career, these all during my early days of PhD, and then those days Philip Maney also, brilliantly, they were at Oxford, the math department, they all helped me at various ways to the basic early career mentors. And then I want to thank my funding agencies from the beginning, last 20 years, Department of Science and Technology, New Delhi, Council for Scientific Industrial Research, New Delhi, Biotechnology and Biosciences Research Council, UK. This is like BBSRC, like NIH of UK, and London Mathematical Society and Ministry of Health, New Delhi. So these are some slides are there at the end. I'm not going through that. They are, just general steps needed for a mathematical modeling. Thanks for your attention.
Thank you, Dr. Rowe. That was a fantastic talk. I will just give make a couple broad comments. So what I loved about your talk is that you highlighted the practical benefits of these models. That's the challenge when you work at the CDC, is taking models like UFIT and telling policymakers that they're relevant and important. So I think you did a really great job with that. Another thing I'll say just generally for all of us, we're all focused on COVID right now, but eventually Dr. Rowe will be coming back to hopefully do some hepatitis C modeling with our division. Him and I are doing some hepatitis C modeling as we speak. And I will also say this, just making a general comment, we're all learning, all the modelers, all of us are learning about this natural experiment we're going through. And this natural experiment with COVID is actually going to feed back to things like hepatitis C, hepatitis B modeling. So it's the equivalent of intensive training on collection of observational data that is going to have impacts on many different diseases. So I'm really excited about it. Last thing I would just want to say is I'm very happy Arni only lives a 2-hour drive from me. So I'm grateful that he gives me time and teaches me the things he's been teaching me for the last 5 or 6 years. So with that, I am going to try to field questions. So this is my first time trying to do this. We have 100 people on the call. So trying to do this might be challenging, but I'm going to look at questions and try and do them in order and feed them back to Arni. I know some people are up against a hard deadline. We just hit 11:02 in Eastern Standard Time in the United States. So we're going to try this and see how it goes. Arni, do you have anything to say before I start fielding?
Fine. Should I stop sharing or should I continue sharing?
Why don't you just, why don't you leave your picture up? I think everyone can see your picture. And let's see how many questions we have up here. All right, we have a couple. All right, let's see. There are some questions. So I'm going to read one from Lori Barker, who works in our division, who is, I will just say, a great statistician and has done a lot of great work in our division and elsewhere. So she says, thanks for the really fascinating presentation. She asked for the isomorphic comparisons of predicted and observed networks. Is it possible that prediction would perform better, or if it would be possible if each node was defined as a small group of 2 to 10 people? It's a long question. And the prediction was whether any of the small groups in the node would become infected by time 2. The question is prompted by practical considerations of efficiencies of testing pooled specimens. And then I'm going to continue what she said. Challenges would include how to handle different networks for people in the same node and perhaps having different characteristics in the node specific cylinder of the socio-demographic characteristics. Or would another approach you presented be a better choice? So that's a long question, Arni, but I think you have the feel for it, right?
Yeah, this is a brilliant question. I like the question because she's right, because when we have the 2 time points, but you have to take the small sample, the machine, the algorithm to match becomes much easier.
Okay.
And then
So,
time taking would be much quicker so that, the matching can be done. Yeah, it is possible to do with the small samples also. Yeah, it's a brilliant idea.
okay. The next question is from Ekta Narwal, who says, which artificial intelligence model was used? So I'm not sure which one I think he wants some specificity on which artificial intelligence model you used for your presentation when you talked about the COVID artificial intelligence model.
Yes, COVID artificial. Yeah, and we developed the model ourselves for the matching, the identifying cases based on the flow charts provided by CDC. Though by February, that time point, using that the various steps involved with all the logical constructions. So it's a reflexive way of handling the algorithms.
Yeah, and we're going to put this recording up and share the slides, and we might want to include a reference section or something like that. A person named RUI, I don't know if that's a first or last name, said, great presentation. Can you talk a little bit about the software you used to build the models?
Well, the models, initial phases, some of the models are based on the C long ago, and then on the Mathematica model makers, the various, they are the, I sometimes use Excel sheet also, some of the data I use Excel sheets.
Well, that's good to know, because I'm a big fan of Excel spreadsheets, and there are people that I work with who do modeling and do simple things and not so simple things in Excel, and it makes it much more accessible to other people. All right, so Rajiv Kumar asked, have you considered any model taking India data? And he says OG, so we have, the problem is the questions are moving. Okay, taking India data, OG COVID spread. So I'm not quite sure about the question, so I'll repeat it. Have you considered any model taking India data, OG COVID spread? So it sounds like, have you been using India data to model your spread of COVID virus?
Yes, in one paper, what we considered in the month of March, whatever the data from the Ministry of Health website available, we use the data for that part of the time, whatever data available, we used it, yes. For India data, we published also.
Okay. Cynthia Reese has a good question. She said, great presentation. How are the modelers and researchers in COVID-19 research handling multiple testing by the same individuals? For instance, there's a talk about the positive to testing ratio, but does that consider unique test results or total results?
That's not total result, because all are not tested. If I understand correctly, because
I think some of it's about multiple testing. So what about the same individual getting multiple tests?
I support that. Because like sports, those individuals who are playing some games, the idea is before the actual game date, they few days before they test,
so yeah,
they quarantine them.
So let me clarify, with the modeling, what happens when you're only getting test results and it's not necessarily deduplicated, where people are getting multiple tests? How will that impact your models?
Oh, I see. Yeah. No, well, that we need to take care. Those who are like a positive individual, say on May 15th, an individual who is positive, and the same individual is positive on May 1st. We need to count them as one individual.
Yeah, and is there a way to do that? Do we know with our current data sources, are there ways that the modelers are getting?
That's actually a very good question. I don't know. That's people are actually, because the idea is that those who are tested positive, they are all quarantined, and then they are confirmation. They are asked to come for another testing. So whether the second data and the first data, they are mixed together, that's a good point. We need to verify, we need to check. But my guess would be they would not be doing that. They would not be mixing these two. Same individual's data, they treated as same individual positive or same individual negative. I don't think they are mixing these two and treating as two individuals.
Got it. Okay, that's going to be the end of the, someone asked one more person asked a question. So it's from Ismail, said, meta-analysis of models is usually the next step after the proliferation or epidemic of models. I agree with that. We're getting an epidemic of COVID models coming out right now. Would the use of prospective collection of data like testing or tracking provide the mechanism of model/approach/scope selection? How this could be coordinated beyond artificial intelligence. So I believe the general question is, what are we going to do when the CDC has thousands of models and predictions from other people? What would you recommend we at the CDC do when we have 1,000 Arnis producing predictions and they all differ?
Yeah, so as I mentioned, and that's also a good question, you know, that's a very sensible question. Because two models, as I mentioned in the interview earlier also in one of the media, that two models are different if they use different mathematical structures and they're different variables and different, so they can't produce same, unless they are using same data, same sort of modeling structure, they can't produce same results. But then the meta-analysis, yes, I mean the meta-analysis should be done, compared, and then they have to test the sensitivity of these models. I really support that, and I think people are already doing, you know, they already wrote some of the reviews on our work comparing with other models that I think people started doing. Maybe they should do in much bigger level. I support it.
Okay, so now I'm gonna field the next question. I will say people, two participants have raised their hand. I'm gonna get to the people with hands raised, but I am going to answer the questions in the window just so I don't miss them. So Prashant asked, does underreporting get influenced by asymptomatic cases? Any comments? There's a lot of comments here. Let me see if I can get to the people raising their hand. I see Kevin popped in. Is there anything I can do? No. All right, we're going to end the talk. One of the reasons is, for some reason, my computer was not plugged in and it's about to lose power as well. So I appreciate everyone joining us. Dr. Rao, I am so thrilled you gave this presentation. You and I have learned a lot about everything you've been doing with all the discussions we've had over the last week. And you really catered this talk to the right level of people that would show up for this talk. Kevin, do you have something to say? Are you? No. Okay, any last words, Dr. Rao?
Yeah, I want to thank Kevin, you know, for, you know, helping us for setting up the IT. You know, he's doing a great job. And thank all the all the participants, and asking brilliant questions and taking this seriously. And early morning, some part of the US is early morning, some other countries late night. So I want to thank all my collaborators, my host, Dr. Thompson, and the CDC. Yeah, thank you all, and thank my family, you know, all the kids across the world. You know, they're really struggling to, you know, just stay at home.
Absolutely. And Kevin Borden, thank you. He is our IT person. He did a fantastic job.
Thank you, guys. You did an awesome job also, too. It made me look good.
Okay. All right, everyone have a great day. Thank you for joining us.
Thank you. Thank you, Bill.